Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Valerie Arboleda, Associate Professor at the David Geffen School of Medicine at UCLA. Valerie is also a 2023 Toffler Scholar. Today, we will discuss Valerie's interest in understanding the genetic architecture of rare, monogenic, and common complex disease. So let's dive in. Welcome, Valerie.
Thank you so much for having me. I'm really excited to be here.
Yeah, I'm excited too. And I want to just start with, what is monogenic and common complex? What is this?
So this is something that's kind of peculiar to the field, I think, of genetics. In genetics, we think about diseases in two flavors. There's this set of diseases where the inheritance was originally described by Gregor Mendel, in that a single gene is essentially causal for whatever trait or phenotype or disease that you're thinking about. So we think about these are the types of disorders like Duchenne muscular dystrophy, where a single gene mutation is causal to the disease and it's inherited in a specific way. But you can pretty much attribute that disease to a single gene mutation. So it's a single genic gene. The other types of diseases we think about are things like common disorders like diabetes, common traits that everybody has, like how tall you are and what are the genetic contributors to height and weight and body mass index, diabetes and your lipid levels. Those have a lot of different genetic contributors. There's a lot of small effects due to a lot of different genetic variants. So, Deborah, you and I differ by roughly 5 to 10 million variants, right? And some of those variants might be the difference between me being short and, if I recall, you're rather tall. And some of them might be as different things that maybe I have a higher risk likely of diabetes because of my family history, but there's probably a genetic component to that. And that might be different than your risk factors. And every variant, none of the variants in common complex disorders will cause a disease. So if I have a variant that increases your risk, but it's a tiny increase in risk. So you have to have a lot of these variants where the risk acts in the same direction to either increase your risk. And that happens just sort of by chance. There's some people who just have a higher risk due to a lot of different variants that happen to be in that person's genetic makeup. And it's still not 100% in those cases. I think in a lot of those cases, the risk is higher, but it's not. None of these things, I think, are in any way deterministic.
That's really interesting because so your research lab, it focuses on this study of these functional roles of these genetic variants in these, in the body and these models. And I can see by that description why you study this monogenic and then this common complex, because I would think that there's some commonality there that you might learn, or some new understanding. But can you walk us through this process of how you identify and analyze the variance between these two? Or maybe there is no connection between these two.
So there is a connection, but it's not one that we typically think of clinically. So I'll take you through sort of what we can do for patients who come into a clinical genetics or any sort of clinic that has a focus around genetics or seeing a genetic counselor. Often they'll take an assessment of your risk profile. They'll do a good phenotyping to see what diseases you may have. And often the patients who are coming into clinical genetics clinic are really sick, right? They're sort of at the extremes of what is typically seen even as within the normal range. And you might have a high lipid level, but the patients who are coming in for genetic testing are sort of off the charts, like sort of 2 or 3 standard deviations sort of beyond what is quote unquote normal lipid or cholesterol ranges. And those patients will get genetic testing either for a panel of genes that we know that if you have a rare monogenic variant in this gene, you're likely to have very high cholesterol levels, a specific type of cholesterol level. And so those are sort of the very clear-cut cases because you have a single gene that you can assess. You have a single phenotype that is a single phenotype or multiple phenotypes in the case of like rare syndromic kids, where you can say, this kid is clearly sick, developmentally delayed, perhaps like small for their age, and not able to eat properly. Sort of there's a number of very sort of these very sick children. It's usually fairly clear that there's probably a genetic etiology, and we then test for every single gene that could possibly be associated with whatever phenotype we see.
And phenotype, can you explain what a phenotype is?
So a phenotype is it's pretty much any trait. So a phenotype could be something like how tall or how short I am. So you can have your phenotype can be your height, your weight, your body mass index, but it can also be what age you spoke your first word at, what age you were potty trained at. It can be almost anything. It can be your blood levels, how many red blood cells you have, how many white blood cells you have, your response to infection. So pretty much anything you can think to observe, we consider that in genetics a phenotype. And it could be something binary, either you have black hair or you have not black hair, or it can be something quantitative like the level of LDL cholesterol in your blood or the level of some biomarker. So we think about these as if we have a really coarse generalization for phenotypes of what constitutes disease. And so I often use phenotype because it's a little bit broader. When people start to talk about, not all phenotypes are necessarily disease, right? That disease, the designation of disease is dictated by sort of the medical literature and where we set that threshold. You know, if you're a BMI of 24, you're still within the normal range. And maybe if you're 26, you're considered overweight, right? And so those are things that like change over time, but also it is a phenotype in and of itself, whether or not it's disease or not disease. I wanted to add, so that connection between monogenic and complex, though, is that we often in clinical genetics attribute everything to a single variant, right? And that's sort of the simple explanation. But I think what is true and what we've observed across large families who carry the same variant, and if that family has the same genetic variant, you would say, okay, well, then everyone in that family who has that variant should have the same expression of disease. They should have the same phenotype that we observe in the mom and the grandma and anyone who inherited that particular genetic variant. But that is not true in all cases. And we know that people differ in some of the other parts of their more sort of like common genetic background. And so we know there's an interplay between common genetics and rare disease genetics, and that whatever your genetic background is can modulate how severe that phenotype is or at what age you actually express disease. So I think one that people often know about in I think is more commonly known, are the BRCA1 and 2 variants that are associated with an increased risk of breast and ovarian cancer. And so they say, I think the common knowledge is that your risk is higher, but what actually influences at what age you will progress to disease? Some people, you have an 80% risk. I think it's 80% by the time you are 80 years old, and that those numbers might be slightly off. But what dictates whether you're 80 versus 40? Those are two very different numbers when you have your first occurrence of breast or ovarian cancer. So I think there's a lot of uncertainty still around genetic prediction, especially for complex traits, is that we know that there's an increased risk, but we know that on a population level, we can't really say much for an individual, whether or not you will have early onset disease, or maybe you'll be one of the lucky ones that makes it to 60 or 65 before you have that first bout, even in the context of this awful genetic variant. And those are the things we don't have the tools to accurately predict, but I think we're getting closer as we start to understand common variants and how they interact with the rare variants.
So that's super interesting. So you said that you try to use a broader description of phenotype. Does that broadening go into things like the environment that we may be exposed to, or the social conditions that we may be exposed to, or economics, or even what our diet may be. There's so many things. I mean, are you exposed to plastic more than other people are exposed to plastic? How broad is that? Because I would imagine if you go way too broad, you can't study or analyze.
I mean, we can't follow everyone for every day of their life until, and really get a good sense of those environmental contributors to health, right? I consider sort of your exposures to plastics and sort of all the other things, like the social determinants of health, your access to care. All of those, I think, are huge contributors to disease burden in specific communities. We are not able to take that into account with the tools we have right now. Sort of even looking at all of the genetic variants at the resolution that we can do it at. It's sort of a question of the more variables that you add, the more challenging it is to really control for all of them, because then you need sort of an exponential number of controls. And so it's something that we're not able to do quite yet. I think we are just starting to be able to look at a single variant and then ask the question, how does the rest of the genetic background influence the expression of that one variant? And that's something that still takes probably a cohort of 250,000 people in order to measure that one thing pretty well with some confidence. So sort of broadening out and adding more things to that is probably just going to require larger cohorts. And I think they're coming. And then the question becomes, how well did we collect that information? And how do you aggregate your collective exposures over a lifetime? Because you can have an 18-year-old who has an exposure who lives in a certain area and maybe has a certain set of exposures, and somebody who's 45 and maybe has lived in very different circumstances and then has moved around over the course of their life and had different exposures at every phase of life. So those are harder. I think those are really hard questions. I think they're going to get there as there's some really exciting cohorts that are collecting this information. The UK Biobank has 500,000 people now that are whole genome sequenced, and I believe they're collecting exposure data for all of those individuals, or they have maybe for a subset. And then if I remember right, I think that the All of Us project out of the NIH is also doing something similar. So hopefully, sort of in the next round of data that's coming out, we'll be able to integrate and think a little bit more about how exposures influence expression of diseases, and also how exposures and genetics together contribute to your risk of disease.
Yeah, and we could talk a little later about maybe some of the technology advancements that might help us. But you're right; it just the variables start to become overwhelming. As you were talking about the person that stays in one place their whole life, and then somebody that has moved around, and I moved 27 different times. And so trying to track my
set of exposures over your course of your life is
right. Yeah, that would be
not trivial.
Right. That'd be kind of crazy. But so can you share a little bit about your journey? You're a physician, but you're also a researcher, and talk about like what kind of sparked your interest, and then really, what was the influence to not only be a doctor, but to be a doctor in research? Because I don't know if a lot of people realize that doctors do research, and they keep up with that side of it.
I think I did not take the most traditional path there. I mean, I finished college and I applied to med school, and at the time I had done a lot of research in college. And I think at the time when I was applying to med school, I kind of thought to myself, I really like research, but I don't really know what that path entails. It sounds very uncertain. And I know I like medicine and people and helping people. And so I'm going to apply to med school. I didn't even apply to the traditional like MD, PhD programs that sort of like is your typical training path. So I went to med school. I decided after my second year, I wanted to take a year to do more research. And so I applied to some of these programs. They used to have these year out programs with the Howard Hughes Medical Institute. So I found a mentor. I applied to the program. I was accepted. And so I took a year off to do research. And at the end of that year, I said to myself, I don't really want to go back right now. I don't think I'm ready. I really want to finish this project. I really enjoy what I was working on. At the time, I was working on a mouse model of XY sex reversal. So we had these sort of curious mice that they were XY by chromosomes, but 75% of the XY in the male mice, or the XY mice, were phenotypically female. And we were sort of trying to understand what the genetic basis was of this. I didn't want to stop the project because I hadn't finished it. So I talked to my boss and I kind of said, I really would like to finish this project. Do you think I could stay longer? Do you think I should get a PhD? And so I went and I applied to the UCLA MD PhD program. And they let me in, which I'm eternally grateful for because it really just changed the whole course of my career. And from that point forward, I was in the research lab. I didn't even think twice. I think I ended up doing another 4 years. I stayed in research for another 4 years, and I went back to med school and said, I really like research. I would love to be able to go back, but I see how hard it is to get funding and how hard it is to get research support. And, at the end of the day, there's always that sort of a little bit of uncertainty around, am I going to be able to support a career doing what this is? So I went and did a residency in lab medicine. So I was doing we did a lot of genetic testing, some clinical pathology, thinking about lipid levels, biomarkers. So my expertise is around running a clinical lab, and I'm board certified. I still do a little bit of clinical sign out. But when I finished my residency, I said to myself, I would love to give this research thing a real shot. And I had some research time at the end of my last year of training. So I applied for some grants, and the NIH gave me money. They gave me enough money to start up my lab and to give people at least some confidence that I knew what I was doing, which sort of tickles me now because I'm I really had no idea what I was doing. They were like, here's a bunch of money, go start your lab. And then my department was like, oh, you got a real big research grant. Why don't you start a lab? Here's some, I looked for some jobs, and UCLA gave me some space, and they just said, go for it. And so it was a little bit of trial by fire, I will say. I learned a lot really fast about how to manage labs and people. And you don't really get training, I think, in any parts of academia on how to run a research lab. So, we learned quickly and I got a good group of people, and I still have some of the people who started in the lab. Some of them are still there and doing amazing work. And then I have grad students who have come. I've had some graduate, and I have some graduating this year. And I think what I don't think I expected from this whole process is it's so much fun to have graduate students and to train them. And it's so sad when they leave because they're ready and they're full-fledged scientists and they're ready to go off into the world. And you say, oh my gosh, it's been this long and you've learned so much. And I think my proudest moment today, I had a student who sort of, oh my gosh, we were talking about someone else's paper, and she went through it, and then she kind of said something that I had told her 2 years ago. She said the same thing back, and I said, I told her that, and she learned it, and then now she's telling someone else.
That's awesome. That's awesome.
It's sort of a nice full circle. People, I say things and they actually listen and they absorb it, and then it sort of becomes a part of how they think as a scientist. So.
Yeah. Now you know how your mentors and your coaches felt when you graduated and into having your own lab and getting your funding and all of that. They felt that proud moment too.
Yeah, it's fun. I think no one told me that was something that was fun about being in academia and mentoring scientists, young scientists, but it's one of those things.
How do you think going the research route, kind of having that focus in medicine, going the research route, going back into kind of medicine, where did it give you a different kind of perspective, or is that pretty common as far as the knowledge that you gain?
I think going the research route has given me a little bit more appreciation. And I think I really I can view things from multiple perspectives, particularly when I see what happens in our pathology labs and our clinical labs. I get their goal is to provide the best clinical care they can. And research, at the end of the day, sort of takes away a little bit from that, in that they don't necessarily have the bandwidth to support all the research part. And I sort of understand sort of that perspective. And at the same time, I come from on the research side. I said, if you provide us access to some of this information or data, it would be really helpful to help the next generation of tests. And I think there's this push and pull between the two. And I understand sort of where each side comes from. And I've learned a lot, I think, going through that, because I can speak in both languages and both sort of, there's this sort of like a way. And I think it's interesting to me as a scientist, depending on how people see me, if they view me as a pathologist, or if they view me as a clinical pathologist, or if they view me as a scientist, I get sort of different, we'll say, reviews back. So whenever I have reviews from let's say like study section from the grants that I submit, sometimes they'll say, oh, you don't have enough scientific background, you need more. So I said, I have plenty of scientific background, or you're too clinical. And so it's sort of really like it's hard for people to kind of wrap their head around the fact that I do both things I do the genetics piece pretty well. And I can do the molecular and cell biology part as well, because that's sort of where most of my training was.
So I think you have identified some novel genes. So you've had some discoveries. Can you tell us about what those discoveries are and why are these important?
So as we started sequencing all of the protein coding regions of the genome, so anything that makes a protein, we started to realize that there are in a number of patients who are really sick and don't have a diagnosis. There's no syndrome that we could sort of say, you look like you have Down syndrome, or some sort of very clear clinical cohort. We started to find patients in whom we would see the same type of pretty bad looking mutation in the same genes. And the kids all looked similar as far as they were developmentally delayed, didn't meet all their milestones at the right time. Sometimes they had a smaller head or heart defects. And so we noticed these, and we realized that they're part of the same genetic syndrome. And it's called, it's due to mutations in a gene called CAT6A, which is a lysine acetyltransferase. It essentially is important in figuring out how open or closed your DNA is, because your DNA is wrapped around these proteins, and it's organized in such a way that if you open up some parts of the DNA, you have more access, and you can control what's being expressed and turn a cell into a brain cell or a heart cell. And if it's more closed, that part of the DNA is more closed, you can sort of shut down that program and turn it into a different kind of cell. So it sort of controls the organization of I'll call it a genetic book. So histones are really important proteins, or histone and acetyltransferases are really important in organizing your histones. And we found these mutations in 3 cases that were within, I think, a month of each other. And we identified a new syndrome based on that. We had identified this syndrome, and there was another group that had also identified it. So we published our findings at the same time. And then we went on to continue with this work. So we really started identifying more and more patients. Once we had reported this, other groups and other clinical genetic testing sites said, hey, we saw that gene too. We have a couple patients who we didn't know what to do with this mutation, but turns out this is probably the right one. And so we ended up there was a Facebook group. There were some families that decided to form a group together so that they could all find each other and learn from each other, because no one knew anything except for like the 6 patients that we initially published, I think, back in 2014. And so now that syndrome is also called Arboleda-Tham syndrome, and it's sort of like the naming scheme of a group called the Online Mendelian Inheritance of Man. So they tend to name things based on who wrote the first papers. I tend to like to think of it from the gene name, but I think it depends. As long as everyone knows which syndrome we're talking about, I think it's fair game. And they're all due to mutations in CAT6A that cause the protein to be too short. The vast majority of them at least cause that. And so we're starting to characterize these patients. We have worked closely with the family group. So we've collected blood and skin biopsies so that we can derive cell lines. And we've started to we've already made, we did this 5 years ago. We made induced pluripotent stem cells. And from those, we're trying to model what happens in brain development. So we're really trying to understand how these mutations affect neurodevelopment at the early stages, because we know these kids have developmental delay. And we're finding some really interesting things. I think my student has worked for the last 3 to 4 years to really characterize all the different levels. We're finding connections with metabolism, different types of transcriptomes and RNA changes, and changes also with the protein level. So there's a lot of things that are disrupted when you change this one protein. So it's literally a single protein change that upsets sort of the whole equilibrium of neurodevelopment. And we're working, I think we're close to sorting the whole thing out, but I think it's taken a lot of wilder turns, like things that I never thought I would be thinking about. We're thinking a lot more about metabolism and how histones are associated with metabolism, which is not something that I think I thought I would be studying if you had asked me 5 years ago.
And why is that? Why is the metabolism important and why wouldn't we been thinking about that?
So I had sort of thought of metabolism as something that happens in the cytoplasm. And I think there's two parts of the cell. There's sort of the nucleus where all the DNA is and everything is organized, and you're trying to find the pieces that encode how a stem cell turns into a neuron, and open up those pieces so that they can be transcribed and translated into protein, RNA and protein. And the mitochondria kind of live outside. They live in the cytoplasmic compartment, and we sort of think about them. And I think that view is changing. I used to think about them as sort of like energy producing machines. They make your ATP, they give you energy to make all the other substrates. And they're also important for organizing the DNA in the brain, because a lot of those enzymes that I'm talking about that add acetylation and open and close the different parts of DNA, they require energy in the form of ATP or different reducing substrates. So there's an interplay there, but there might even be a more direct connection, because a lot of the parts of the biochemistry cycle are important for they provide that piece that adds the acetyl, that's the function of KAT6A, that acetyl group that's important for adding those acetyl groups to the histones. And that acetyl group is provided by metabolic substrates that go into the nucleus. So, when we think about it from the broad perspective, yes, it makes sense. These molecules are so small, they don't have like a special transporter. They just kind of like float through. But I think we had always just been focused so heavily on the nucleus. We hadn't thought to assay metabolism in a meaningful way. And people have done metabolic studies in these patients, but you really need to, if you're doing it at the organismal level or at the human level, like I'm taking some blood and asking what's your metabolic state, it's very dependent on what you ate over the last 24 hours, how much you slept, what time of day it is. So if you haven't controlled for all those things, it's almost impossible to get a good readout of what's uniformly different in these patients relative to others. If you'd look at their numbers and you'd think, okay, these are just in the normal range. And that's what most of the studies have shown. They have not shown that they are significantly different, but no one's done the right study. This is the way I think you have to do it properly.
Yeah, that's interesting. So how rare, you said there was 6 patients that started. Do you have more patients? How rare is this?
It's rare. So we are looking at probably somewhere between 1 in 50,000 to 1 in 100,000 births. Right now for the KAT6A Foundation, I think the last time I checked the website, they had probably 300 to 400 patients who had been had a genetic diagnosis of a mutation in KAT6A. Those numbers are probably an underestimate, because the only place where a lot of this genetic testing is done are sort of in high resource countries. The US, the UK has sequenced all of its kids with developmental delay in some large scale research projects. But I would say the vast majority of kids, unless your parents are advocating for you, genetic testing might not ever get to you. And even if it does, you may not find the family group to like add yourself to the list. And then there's lots of parts of the world that have no genetic testing. So they may not sort of come into the folds of groups that are advocating for these patients and their families.
But I would think that it's important to study this, because it's somewhere maybe in the future, just the knowledge that you're gaining now, it's rare today, but maybe it's not as rare in the future because you have more information, which then allows for more attention and money for diagnosis and treatment. Because when it's really rare and there's only a few, it's hard to get the attention for, unfortunately, that's just how it is. But that's why it's so important to continue, I would think, I don't know, you tell me, to do this, because something that might be rare today may be somewhat common in the future because you've done this work.
So 100% agree. It's really hard to get funding for these sort of rare, what I just described as a rare pediatric syndrome, right? There's 300 cases in the US. People aren't really running to fund it because they're like, well, you can only really help 300 people. I think to push back on that argument, and it's not even that we might find more people with disease. I'm not sure that's the way I would look at it. I kind of think about it as these genes have really critical functions in human development. And they're all known, or many of them are known to have critical functions in disease progression. So CAT6A is known to be a partner in a translocation. So it's a specific type of genetic mutation where two genes that aren't supposed to be joined together are actually fused in certain types of leukemia. So there's a sort of advanced leukemia that can happen with the fusion of CAT6A with other binding partners that causes it to be overactive. And the other place where people have found that CAT6A is actually really overexpressed in certain types of breast cancer. And so there, I think, are phase 1 clinical trials with CAT6A inhibitors, trying to see if those will help in sort of as an adjuvant therapy for patients who have breast cancer, specific subsets of breast cancer that overexpress CAT6A. There's another disease that we study that we didn't describe initially, but we work closely with the ASXL1 Foundation, in which mutations cause Boring-Opitz syndrome. And that one's a really nice sort of like juxtaposition of a really rare syndrome. But this particular gene, ASXL1, is actually a driver mutation for a lot of myeloid leukemias. So they're found in somewhere between 20 to 40% of myeloid leukemias. And when you see these mutations, they actually confer a worse prognosis. So there's a real case to be made that we should be studying these patients because we don't understand even how this gene works. We've been studying it for 5 years, and we don't have an antibody that can even, we think, really accurately detect the protein. So we're sort of stuck at this level where we can see that it has effects. We've done all these studies. We see these downstream effects. We have no idea what the protein, like what size the true protein is and where it sits, and sort of like being able to see it even in a cell, because the antibodies, we've tried all of them. I've spent more money on antibodies than I care to admit. And we just can't detect this protein at the cellular level. We can't see where in the cell it is expressed. People tell us where they think it's expressed from some protein atlases, but this protein is really critical in leukemia and in these patients. Maybe you're not going to cure the patients because their disease is pretty well progressed, right? This is a development. So they have this mutation from before they were born. And so it's hard to fix some of those early developmental timelines to make a normal brain. But maybe you could fix or alleviate at least some of the suffering or some of the cellular issues that are occurring at the like differentiated level. Like maybe that you could sort of make some things better, even if you can't fix everything. But for the patients who have cancer, those are continually coming up. They're being found in clonal hematopoiesis, which is sort of a precursor to cancer. And they're literally the exact same types of mutations. Like you put them side by side, and we've had patients who are diagnosed who have like sort of early stage, we call them clonal hematopoiesis. So we can see the cancer starting to become a clonal thing, but it doesn't look like cancer under the microscope quite yet. So we follow those patients closely. And sometimes their genetics people will say, hey, we think that this is actually Boring-Opitz syndrome. And our clinicians are like, there's no way this 70-year-old has Boring-Opitz syndrome without being diagnosed early. Like all of them are diagnosed at birth. I think there's a lot to be said. Like if you study rare diseases, it's going to provide you insight to a lot of really important diseases that we don't have any therapies for. We don't even really understand how it functions in blood stem cells or why it causes cancer and why it doesn't cause cancer in the kids, right? These kids, they don't have the longest lifespan. I think the oldest ones are around 30 right now, but they don't have leukemia. So there's something different about sort of the bone marrow niche but also sort of like that if you have all your cells have the mutation versus just the cancer cells, and then the rest of your bone marrow, all the supporting cells in your bone marrow are maybe saying grow, 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 and your stem cells don't have any way to say stop growing. So they just keep growing until they're just taking over everything. So I think there's a lot that we can learn from the boring opens patients, but they're rare. I think they're even less than 300. There may be 100 cases that we know of in the US.
Wow. Wow. You just used a term that I don't know what that is. Not chromomatic, these modifiers that chromatin.
Mm-hmm.
What is a chromatin modifier?
So a chromatin modifier is a class of genes that we study, and the whole goal of these specific genes, when they turn into proteins, their goal is to open and close the DNA, or like to identify regions of DNA that they want to open to transcribe and make into RNA and protein. So chromatin modifier is anything that opens, closes, or reads the DNA. And it's called chromatin because DNA is like a string, and there are little proteins called histones, and we wrap the DNA around those histones to kind of organize itself into a nice more compactly, and that is called chromatin. So the combination of your histones and the DNA wound around it is called chromatin. And so we call them chromatin modifiers because it sort of affects how open and closed that chromatin is, and by default, how much DNA is exposed.
Okay. Yeah, that's very helpful.
So chromatin modifiers, and I think the other word you were saying was chromatinopathy, which is the diseases caused by mutations in chromatin modifiers. So if you have a mutation in a gene like KAT6A or ASXL1, those are all considered chromatin, sort of the bigger umbrella of chromatin modifiers. And we started calling them chromatinopathies because we know that they cause sort of these rare pediatric conditions.
So that's the class of these modifiers that you're studying. Okay. Exactly. So really you're because this is kind of these are new and you're studying, it's really building a taxonomy. It sounds like you're building a taxonomy for the future for these different kind of, this different understanding of the genetics of some of these people or the mutations, so you're really kind of laying that foundation for future research.
We're trying to, because I think there's a lot of reviews because a lot of people know that these cancers, pediatric syndromes that are like neurodevelopmental syndromes, there is a known enrichment of chromatin modifiers that are mutated in both of those settings. But actually trying to pinpoint what these genes do, they can be fairly hard to study. They tend to be rather large in size. They're more difficult to clone. And it's sort of an interesting fact. So they're harder to just take that gene and put it into a bacteria because the bacteria die if you overexpress it. And if there's any what we consider leaky expression, the bacteria say, we don't want to grow this. I know you want this DNA val, but we're not going to grow it. It's killing us. And literally the bacteria just die. So we've actually, and we talk to plant people who study the same homologs in plants, and they say the same thing. They say we have to have special conditions for some of these genes because they tend to be just toxic if you have too many of them, and you have to really carefully regulate their expression. They can't have too much. It's kind of like Goldilocks, not too much, not too little. It has to be like kind of just right in order for the cell to grow properly and differentiate on time.
So none of this, and maybe I'm wrong, but none of this research was being done before we kind of sequenced the DNA, because this is pretty new science. When we think about science, it's just in the last couple decades.
Yes, I don't think we had the full genome sequences of KAT6A, ASXL1, or if we had them, they were difficult to identify in people because these genes are so long that you wouldn't sequence them unless you had a reason to. Once we started doing sequencing on a clinical basis and we said, you know what, let's, instead of sequencing the 10 genes we know about, let's just sequence everything and see if we find new things. And that's kind of exactly how we found KAT6A. And I think around that time, 2010, 11, 12, everybody was sequencing all of their patient cohorts. They said, we have this bank of DNA from all the patients I've seen that I am like, I don't know what they have, but I think they have something similar. And and the community just identified hundreds of new disease-causing genes. And sometimes they found people who had two cohorts that appeared totally different, that had mutations in the same gene that were in different parts of the gene, and they looked different, even though it was the same exact gene. And so a lot of surprising things really came out of being able to sequence everyone and to sequence everyone thoroughly. And, at the end of the day, when I think about it, there was so much sequencing done, and it was on a version of the human genome that still had a bunch of missing pieces. Like we hadn't truly finished the genome, right? We said we finished it in 2003, and now we're really starting to get to those ends and the pieces that were hard to sequence by the technology back then. The newer technologies now really have made it possible to fill in those gaps, but also to better understand the differences between different ethnic groups and different ancestries, and figure out, like, okay, are these more common? Is this a region of the genome that is duplicated in one group but not the other, and just leads us to be more accurate in some of our detection methods.
So, that's very exciting. What is the most exciting thing that you think that's going on right now in your area? What are you most excited about?
I'm really excited about the linking of genetic information. So a lot of our cohorts, we build these biobanks, and we've linked genetic information, which in and of itself is interesting. But I would argue that having a lot of genetic information is really meaningless unless you've linked it to clinical phenotypes or any phenotype. It doesn't really matter to me what phenotype. So now that people have started to build and sort of governments have decided that this is a priority, we want to be able to understand genetics and genetic medicine, and maybe we can improve human health by doing this. But it's not something that like a single investigator like myself can do. It's like you need the buy-in and support of big government entities to say, okay, we are going to do this systematically. We are going to collect all this information. We're going to put it all in one nice structured database. And then we're going to sequence everyone and give you that link to the genetics and the phenotypes, so you can look for these associations. So you can identify which genetic variant might be causing which disease. And so that's probably been one of the most exciting parts, is that we can start to look at this in diverse individuals and a lot more people, and have the power to look for very small effect sizes. Because I would argue we can see the big effects. We can see like the genes like CAT6A, ASXL1. When you just mutate one copy, it's devastating for the child, right? It's a horrible, the effect of the gene mutation itself is just so obvious, and it's apparent at birth. But there's so many other ones that we kind of say, we're not sure. This could be something, this could not be something. And those ones are kind of the ones that are the most harrowing, because if you're a patient, we can't give you a real definitive diagnosis. We put this into this like catch-all category that we call variants of uncertain significance. And then we hope that someone else figures it out in the future, so that we can come back to the patient and tell them whether it's benign and not causing disease, or if it's causing disease. And we're kind of still in this stage of clinical genetics where it's pretty coarse. Like we have three categories. It's bad, it's nothing, and we're not sure. And the not sure category is huge right now. So we're slowly chipping away at this. I think biobanks has been huge in helping us get to that point. And I think the biobanks are just starting to like really mature in size and in sort of the depth of the phenotyping. And then the second piece that I think is really exciting is the use of AI. So there's a lot of these language learning models that have really fed in a lot of data and then asked sort of agnostically in an unsupervised way, what makes a certain mutation bad or good? And they're able to, some of them are really good at predicting whether something is going to be bad. And then they can also distinguish between something has, like we call it a loss of function, as if you just got rid of the whole gene, or gain of function, where there's like a new acquired function of that gene that didn't used to be there. And that's what's causing disease. And previously, those two were sort of lumped together. You couldn't distinguish between them in pathogenic versus non-pathogenic. But some of the newer ones are actually able to distinguish between the two, which I think is probably one of the coolest things I've seen in genetics recently. So I'm excited to like see how that integrates and go back to some of our older data sets and ask these questions around, hey, we saw those variants of uncertain significance. Can we ask whether any of them fall into these categories in these new language learning models that are AI generated and are publicly available. So that's the other sort of nice piece about genetics is at least in the academic sector, there's a big push to keep things open source and let people discover what they can from this data, because no one group can really do it's so much data, no one group can do it themselves. So it's a really exciting time to be a geneticist. More data than you could possibly imagine. And maybe the biggest hurdle is computational power.
Yeah. And time. And time.
True.
Yeah, as you were describing that, I was thinking about the patient side and how unsettling that would be to, bring your child in or have these conditions, and there is just no answer, like uncertain. We're just not sure. And I think that has to be, very frustrating. And so, I mean, your message of what's exciting is one of hope that, because sometimes bad news is better than no, we're uncertain. We don't know. Right.
Exactly.
And,
I think the uncertainty can be really frustrating, right? We went from one diagnostic odyssey. So we used to say like patients didn't have a diagnosis and then they got genetic testing and then they had a diagnosis. But then there's always a handful of people who get genetic testing and come out with the variant of uncertain significance. And I think I agree with you. I 100% think that an answer, even if it's a bad one, will at least it provides you a community to which you can go and ask questions and get sort of some information. But a variant of uncertain significance sort of deprives you of like, not really sure you should be in this community versus being in a different community of like these sort of small groups of rare diseases where they really rely on each other to provide a source of comfort and hope and sort of like, this is what's in store for the future. This is how we've managed this particular challenge.
Yeah. So do you think these are the key kind of challenge, the computational power, time, funding, a lot of stuff still isn't open source. A lot of it isn't open data yet. Are these the big challenges that is creating kind of a gap? Or what are those big challenges for these genetic discoveries that, are just really important?
There's a handful of challenges I think that remain unsolved. Some of them are sort of infrastructure based in that, we do a lot of research in these rare disease communities, and depending on, we always have IRBs and sort of do everything to the letter of the law. But the families we work with, they're like, we have a rare and devastating disease. We don't know how long our children are going to live. Can you please work faster or get this done quicker or move this to like another institution more quickly? And the IRBs are very, at least our IRB at UCLA are very conservative around genetic information. They don't know they're not as attuned to sort of like the power of genetic testing or genetic information and whether it should be allowed to be more freely shareable, because I think some of the challenges are that you need genetics linked to phenotypes. And in the absence of that, having genetic data is just like having 4 letters repeated over and over and over again, and it doesn't really matter what it says, right? And to make those sort of inferences and associations with disease, you need them linked in a meaningful way. And we need to be able to share it because you find things in the larger cohorts. And I think there's been a lot of challenges, at least sort of in my experience, around trying to share what we've discovered easily with other people or trying to develop protocols in which we can share freely, because a lot of the most widely used tools in genetics are ones that you can just easily download off the web without firewalls and all these extra protections. But I think a function of this is that we don't have good laws and protections around sort of genetic privacy. And so it's sort of like a back and forth. Like we need to set it up so that we as a community can share this work freely. And I think that's what at least in the rare disease community, I think that's what they want for us to be able to do. And we need to make it sort of clear, like that is the goal. And this sort of idea, we're always protecting the participants. Our participants want to be protected, but they want you to share. And I would argue it's almost impossible for me to do that. Like my IRB won't accept that I should be able to sequence someone's genome. And then if they say, okay, we can put it up and share it easily with my friend at the next hospital without having to go through like a 10-week process. So I think that's one of the big challenges to me in rare disease genetics, is that there's sort of a disconnect between maybe what the patient wants and what we think about patient protection. And that infrastructure is not solidified in any meaningful way. It's sort of like at the whim of each individual institution's IRB.
Yeah. And IRB, I think it's what is the I for? Because it's research board, right?
It's an institutional review board.
Review board.
So their goal is, and it's a goal that's much needed. Like they need to protect participants in research, but genetic research is a sort of space that everyone's worried about from the protection. Like in theory, you could identify people if they're sitting in a database in theory. And I think it's possible, but I would never go through the trouble unless I really had vendetta, I suppose. But it's a lot of work, right? It's a lot of work, and I'm not sure even I could do it on my own. It's sort of gone beyond, I think, even what the rare disease community members want. Like they want me to be able to share. I want to be able to share all of the things that we do after we publish them. And it's a lot of paperwork. And I would rather be thinking about the research than filling out a lot of forms. I mean, I will do it, but it's certainly not my favorite part of this job.
Yeah, no, and that's fair. And I think there is questions as we gain more and more information. Where is that privacy? Where is that, that protection? Where is the ethics and the ethical use of this information? And if we just go to the questions that social media and our IT giants are trying to answer, it's
not an easy question. I think this research infrastructure was not made for this day and age where people are misusing information, particularly genetic information. And at the same time, I'm a little bit of an optimist in that I feel like data and science moves faster when we work as a team and we work collectively, and everyone works at sort of the things that they have the capacity to look at and think about and what they're interested in. And I don't want to be the sort of that barrier. I don't want to be that person who has to do it here because my IRB won't let me share to a certain place. And I think that's, it's a solvable problem. It sort of requires sort of like buy-in from multiple groups.
Yeah. Well, this podcast is kind of focused on brain science and the future of the brain. And do you have any thoughts to share there? The connection of some of the science that's going on in the brain with the body, and just any kind of thoughts about the future of brain science?
Brain is a very complicated organ. And I think it becomes evident when we're trying to really model brain relevant disorders in any model system, in mice, in cerebral organoids, in cells, like trying to think about how these are affecting the brain, because the brain has sort of its circuitry, that some of which is genetically acquired, but there is the component of there's a stimulus from the outside that is also sort of helping rewire and connect pieces of your brain based on what you're seeing and what you're hearing, and sort of all that sort of external stimuli, sort of this environmental component that we talk about but have a really hard time quantifying. And so I think the brain is sort of the pinnacle of this, right? We have to understand sort of how it's built and how you create these circuits. But I think that the last piece of brain science, or the part that's going to be the hardest to really quantify, is going to be how that external input influences the circuitry and the gene expression and sort of all the different, like how the brain interprets the signals it's seeing, and how the genetics helps you interpret how it's being seen, right? There's nothing about genetics that's deterministic. We always see cases where we think this should be a disease and the person's walking around just fine. And so there's always exceptions, I think, to every single rule. And the question is, why is there an exception? Is there something in the genetics, something in the environment, or something that's just, so there's so many options once you come to sort of like the ingenuity of the human cell to survive. I think to me is like sort of the most fascinating thing. And I think the brain is really the sort of ultimate integration of everything, genetics, environment, over long periods of time. So I think we have a ways to go, even in understanding the genetics of brain development, right? Before you have all that as much environmental input, right? You think about just the time of fetal brain development, which is where most of our work is. There, you don't have that external stimulus of like seeing the sun and other objects, but you do have the environmental stimulus of the maternal milieu. So it just gets more and more complicated.
Well, and that's going back to your comments about collaboration and different communities coming together to share what they've learned. You have people that are studying environmental impacts on people, but you have expertise in trying to understand the brain. You have expertise in the genetic and the mutations. And if the communities don't come together to try to understand and share, it will lead us to, just kind of slowing down the process of really trying to understand what is going on with ourselves and how things, keep us healthy or make us sick.
Yep. I mean, yeah, collaboration is sort of the only way things move forward, because I certainly can't be an expert on everything. I don't really want to be an expert at everything. I know what I know, and we've had really fruitful collaborations with people who are more of I think, classically trained in brain science and neuroscience than I am. So I've learned a lot. So I think that's maybe the other piece, is that I think I'm a lifelong student. I really like to learn new things, and sort of delving into the brain science of neurodevelopment in these particular disorders has been a really sort of it's been a wild ride. It's been fun.
Yeah. So you look out 10, 15 years from now, what do you see, or what do you hope to see for the future?
I really hope that we have some treatments for these kids. And sort of by extension, if we have treatments for some of these kids, we may have treatments that will work for some of these cancers that also have the same mutations. I think the communities that have spent so much time and given literally blood and pieces of themselves so that we can study these mutations better, I think we can get there. And I don't think it's going to necessarily need a gene therapy, although that would be amazing. I think we can find things that'll make living with the condition better, and perhaps like sort of improve, sort of speed up some of the development of things that are there. Because at a young age, I think a lot of these kids still have some plasticity in their brain. And if we can sort of jump on that at an early stage, I would love to be able to see to a small clinical trial, right? There's only 300 patients. So you could, in theory, get to everybody, and it would sort of just be amazing to be able to impact the lives of all these patients. So that in 15 years, I mean, I couldn't have predicted COVID. And when I started my research lab, if you had asked me if I was going to be doing COVID testing, I would have been like, no, I don't do infectious disease. But here we are, and we're still doing our rare disease research, but we just do a little bit of infectious disease work as well.
Yeah. And who knows, we may find a connection there in the future.
Yeah. I would. I'm beyond trying to say I can tell you anything beyond what's going to happen in the next hour. So
right, yeah, that's definitely fair. I want to just go back really quick to your excitement about your team and the mentorship and just the big surprise about how wonderful that is. Do you have any kind of words of encouragement or coaching for those that might be listening that are thinking about or in the middle of their journey becoming a clinician / researcher or wherever? Any thoughts?
Yeah, it's a long road. And I tell this to a lot of my students who are in the MD PhD program, that this is not a sprint, it is a marathon. And so you want to pace yourself. Don't stop living your life in order to focus on your career, because they in some ways, if you can get it to happen sort of side by side, it's the ideal scenario, because there will never be a right time for anything. Your personal life, often, the type A's in medicine, we tend to sort of prioritize career and just say everything else will come later. And I would sort of argue, like, I think it's really beneficial to sort of your mental health and longevity within science and medicine to have a balanced life. And I don't mean balanced in that you can do everything all at once. I think more just to have time to yourself and with family or whatever, sort of where you find comfort, and then also time to do your science. And I think it makes you a better scientist if you have both pieces back and forth. And it doesn't mean, and I work a lot, and then sometimes I'm on vacation and I'm on vacation. And I think those are the pieces of, it's really a marathon, and it's easy to burn out if you take the road of, I just have to get to this next point. I just have to get that R01. I just have to get tenure. I just have to get that next job or that next source of funding, because there's always going to be something else to do. So it's really important to pace yourself and sort of decide at some point what is worth spending all that extra time on, and when it's a good time to, like, when it's time to take a break, just, you have to take it, because no one's going to force you to take a break. You're just going to have to sort of set those limits yourself.
Yeah, I think that's very, very wise. Valerie, you're just a special person, and your passion and your excitement of what you do is just absolutely wonderful and pretty contagious. So thank you very much for your time today. How would people reach out and contact you if they're wanting to collaborate or find more, or even, if, I don't know, if people in that, they just haven't had a diagnosis and they may be one of these 300 that have a rare disease. How do people get in contact with you?
The best way is usually by email. I would say if I don't email the back the first time, sometimes the second shot is worth it, because just things get buried. But my email is on my website. It's on www.arboledalab.org. And you can also find me on the UCLA website. And I think my email is on there too. So I do routinely get emails from people who are interested in being part of our research. And I think on our website, we also have a contact me, so you can sort of submit a form and it gets to me that way also. So any of those ways, I'm happy to hear from people or comments, and also people are interested in the science. I love to talk about it.
That's great. Well, again, thank you so much for this passion and this energy and your dedication, and just the time you're spending learning and pursuing the breakthroughs in the science. So thank you very much for your time today.
Thank you for listening, and thanks for chatting with me. I always love to talk about science. It's one of my favorite things.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.